So we have already seen that in CHF, heart is not able to pump the blood. Now, how will it be able to pump the blood? If we increase force of contraction of heart. So to increase force of contraction, we will see these two classes of drugs, cardiac glycosides and sympathomimetic drugs. Apart from this, we will also see these drugs not in this topic under CHF topic, but in hypertension and diuretics topics. Now, why these will be given in CHF? Because these will reduce the preload and afterload in CHF. So let's start with cardiac glycosides, which is the most important class of drug under CHF management. Now, before going into cardiac glycoside mechanism, please see the action potential video so that you know what is action potential, how myocytes are contracting, and then only you will be able to understand mechanism of cardiac glycosides. So in generally, how does myocyte contract, that we will see, and in that how cardiac glycoside is blocking or increasing force of contraction of heart, that we will see. So in general, at resting state, too many sodium and calcium ions are outside the cell and potassium ions are inside the cell. So this is the starting phase. Sodium and calcium out, potassium in. The resting potential is minus 90 millivolt. Now, from pacemaker cells, few sodium ions enter inside the cell. This increases potential slightly towards positive because sodium is positive ion, up to minus 70 millivolt. Now, this minus 70 is the threshold potential at which voltage gated sodium channel opens and too many sodium ions enter inside the cell. Now after this, along with sodium, calcium ion channel also opens, L type, which enables calcium entry inside the cell. Now because of positive ion entry inside the cell, sodium and calcium, the potential moves towards plus 40 millivolt. Now at this point, since too many positive ions have entered inside, some positive ions have to go out to compensate the potential, and that's why potassium channel opens and it throws some potassium ions outside the cell. Because of this, there is early depolarization. The potential slightly reduces, comes towards negative. Now, if you see the calcium channels, it is not yet closed because they are slow channels, and that's why there is a current which is set. So calcium ion enters inside and potassium ion goes out, so simultaneously. So this is setting the potential constant and there is a plateau in the action potential. Now after this, now till now what has happened, potassium has gone out, sodium has come in and calcium has come in. But at resting phase, in naturally what should happen, sodium should be out and potassium should be in. So to reverse it, to bring the resting state, generally in action potential, this sodium potassium pump opens, which exchanges sodium and potassium ions, throws sodium out and potassium in to bring resting phase. But this pump is blocked by cardiac glycosides, which is the mechanism of cardiac glycosides. And by blocking this, what is happening now, sodium is retained inside the cell and potassium is still out, but sodium has to go out. So how sodium will go out? There is another channel which works here, which is known as sodium calcium exchanger. So in this, what is happening, three sodium ions are thrown out through this channel and pump, and one calcium is entering inside the cell. So this is how it is sending to sodium ions out, taking calcium in, and potassium ions are also coming in through some other potassium channel and or pump. Now here if you observe what is the different thing that is happening because of blockade of sodium potassium pump. If this was open, then there was just exchange of sodium and potassium. But when this is blocked, there is exchange of sodium with calcium. Calcium has entered inside the cell. And calcium is important for contraction, as we have seen in action potential video, that the more calcium ions present inside the cell, the more contractile protein troponin activation will be there, and more actin myosin activation will be there, which will lead to better contraction of myocytes. So this is how, by increasing calcium concentration inside the cell, cardiac glycosides are increasing contractility of the myocytes. Let's see some examples of cardiac glycosides. So in cognosy you must have come across digitalis plant, which from which actually this cardiac glycoside is extracted. Now this digitalis plant, it is of two types, digitalis purpurea and digitalis lanetum. From digitalis purpurea, digoxin and digitoxin cardiac glycosides are extracted, and from laneta, lanetoside A and lanetoside B are extracted. Let's see the chemical structure of all these cardiac glycosides. The structure of cardiac glycoside consists of two parts. First, the sugar part, glyco, which represents sugar, and the second part is non-sugar, that is aglycons. So, glycons plus aglycons is the structure of cardiac glycosides. Now, let's see first the structure of aglycon part, and then we will see how sugar part is attached to this aglycons. This is the skeletal structure of glycons in cardiac glycosides, where you can see there are four rings A, B, C and D. All these are saturated. There is no aromatic ring. The A, B and C rings are 6-membered rings, and D is 5-membered ring, all attached to each other. Now, apart from this, the four rings, there are four substitutions that we need to remember. There are the two hydroxy group, one on 3rd carbon and second on 8th carbon. So these two hydroxy group and two methyl group, one on 10th carbon and second on 13th carbon. So these are the four substitutions. Apart from this, the fifth substitution, alkyl substitution, varies in different glycons structure. Now if you see, this is the digitoxygenin glycon. In this, everything remains like skeletal structure, 2 CH3, 2 hydroxy group. The alkyl group is a lactone ring. So this is digitoxygenin. Now let's see the second aglycon aglycon structure, that is digoxygenin. So in digoxygenin, again alkyl group is lactone, but what is the difference then? There is one extra hydroxy group on this carbon. If you see in digitoxygenin, there was no hydroxy group on this 12th carbon, but there is extra hydroxy group on this 12th carbon in digoxygenin. Similarly, in getoxygenin, there is one extra hydroxy group on this 16th carbon. Rest everything, 2 methyl group, 2 hydroxy group, is similar to digitoxygenin. There are two more glycons, auberginin and strophanthodin. So here, instead of methyl group, it has been hydroxylated. It has become CH2OH, and there are two more hydroxy group here and here. In strophanthodin, this CH3 becomes aldehyde group, CHO. So 5 glycons we have seen: digitoxygenin in previous slide, degoxygenin, getoxygenin, oabagenin and strophanthin. In sugar part, we have 4 sugars. Taking example of glucose. So its structure we all know the basic structure: 6 carbon, where there is one CH2OH outside the ring structure. If you see other sugars, that is digitoxose, rhamnose and cymarose, all these three they contain CH3 instead of CH2OH like in glucose. Apart from this, in digitoxose there is this OH missing from this carbon. In rhamnose, again this OH is there, but the orientation of this OH is changed. It has come down. So that's why it is beta L rhamnose, not D rhamnose. Then in cymarose, if you see, it's beta D cymarose, where instead of this hydroxy group, it has become OCH3, methoxy group. So these are 4 sugars: glucose, digitoxose, rhamnose and cymarose. So let's see the structure of one of the example of cardiac glycoside, that is lanetocyte A, which is extracted from digitalis lanata. It contains a glycon part, digitoxygenin. So this is the structure of digitoxygenin that we have seen already. Now this digitoxygenin is connected to sugar molecules through C3 carbon, the hydroxy group which was present on third carbon. Through that it is connected to sugars. These sugars which are present in the cardiac glycosides, they are connected with each other through beta 1,4 glucosidic linkage. Why 1,4? Because the first carbon of the sugar is connected to the fourth carbon of another sugar. So beta 1,4 glucosidic linkage. Now there are 3 digitoxose sugar. If you see 1, 2 and 3 digitoxose sugars present in lanetocyte A. The third one is containing acetyl group on third hydroxy group. So this is the third digitoxose sugar, in which this is the first carbon, second carbon and third carbon. Now the third carbon is containing, normally it should contain hydroxy group, but here it is containing COCH3, that is acetyl group attached to this oxygen. So these are the 3 digitoxose sugars. Apart from these 3 digitoxose, there is one glucose sugar also attached. The fourth sugar is glucose sugar. So this is the lanetocyte A structure. Digitoxygenin glycon part, 3 digitoxose sugars. The third digitoxose sugar contains 3 acetyl group and fourth one is glucose. The structure of lanetocyte B and C are almost similar to lanetocyte A. The only difference is the glycon part. So lanetocyte B is containing getoxygenin. Lanetocyte C is containing degoxygenin as a glycon part. So we have seen digitalis lanata cardiac glycosides, that is lanetocyte A, B and C. Coming to digitalis purpurea, from purpurea we get 2 cardiac glycosides, digitoxin and digoxin. So the structure of digitoxin and digoxin is almost again similar to lanetosides. The only difference is, few differences that are mentioned is, first, there is no fourth sugar, glucose sugar present in this. There is only 3 digitoxose sugars. Second, the 3 acetyl group present on third digitoxose sugar is not there. It's plain digitoxose sugar. Okay, unlike In lanatocyte, here there is no 3-acetyl group. There are 3 plain digitoxo sugars attached to digitoxygenin glycon. So this is forming digitoxin and this is digoxin. In digitoxin, the glycon is digitoxygenin, and in digoxin, the glycon is digoxygenin. So here you can see there is extra OH group which makes it digoxygenin. So with this we have completed cardiac glycosides mechanism of action and the drugs and their chemical structures. In next part of the video we will see sympathomimetic drugs and role of vasodilators in CHF.